So welcome to the class on spinal cord histology, which is the addendum lecture to the large lecture on spinal cord external features and internal features. In this particular class, we are particularly going to focus on the following. We look at a brief topographic anatomy of the spinal cord. We'll also look at the general structure of the spinal cord, of the general internal structure of the spinal cord. We'll then focus on three major areas. One is on spinal cord ependymal lining, and two, on the spinal cord gray matter. We'll focus on the organization and role of the spinal cord gray matter. We'll also look at the organization and role of the spinal cord white matter. And we'll take more time to practically distinguish different spinal cord segments. So starting with the first agenda, which if you listen to the longer lecture is not really a hard concept. We know that the spinal cord provides innervation to the trunk as well as to the limbs. And this innervation is done by the different spinal nerves. So the spinal nerves come from the spinal cord and they ramify. Their branches supply different structures in the trunk as well as in the limbs. So it's important to note that through these nerves, the spinal cord receives sensory terminals from the periphery. The peripheral structures can either be somatic structures, which means that either we are talking about skin or skeletal musculature, or even visceral structures like the digestive system, blood vessels, and the like. After receiving those sensory terminals, it will relay them. But the spinal cord also gives rise to motor neurons, which go to musculature. They could be skeletal musculature, they could be cardiac musculature or smooth musculature, and even exocrine glands, the ones that produce body secretions. So we have both afferent and efferent functions in the spinal cord. This image shows you the different sensory nerves that arise from somatic structures, which eventually get to the spinal cord. After receiving and even giving rise to the efferent impulses to different effectors, the story doesn't really end there. As a matter of fact, the spinal cord also has profuse connections with the brain. The connections that the spinal cord has with the brain are connections that involve axons of neurons, which are either going up. The ones going up are termed ascending pathways or ascending tracts. Remember, a tract is a group of axons of neurons with similar function. You can call it a tract or a fasciculus. And so there are several ascending tracts carrying sensory information to the brain. But there are also several descending tracts. The descending tracts are the axons that relay impulses from above downward to the spinal cord. The descending tracts will tend to have motor functions, while the ascending tracts will tend to have sensory functions. The linkage that the spinal cord has with the brain enable the brain to control a number of functions of the spinal cord, but also to receive the impulses, the sensory impulses from the rest of the body. And so it's important that the brain and the spinal cord are connected for that particular reason. We already know that the spinal cord is located within the vertebral column and it extends from the level of the foramen magnum to lower down at a point we call the conus medullaris. The conus medullaris, that abrupt ending after a small swelling of the spinal cord. The location of the conus medullaris varies depending on age. For example, in adults, around L1, L2 junction at the time that a baby is born, around L3, and perhaps 6 months in utero at around S1. And in the beginning, it actually extends the whole of the coccyx up to the 3rd month. The understanding we have about this is that the spinal cord grows slowly compared to the vertebral column. So the vertebral column outgrow the spinal cord. That's why the spinal cord appears to shorten as we grow. In terms of the spinal nerve roots, we know that the spinal nerves originate in pairs from different segments of the spinal cord. Now, these spinal nerves attach in a linear series of nerve roots. We can see the linear series of nerve roots. Or we have the rootlets, multiple of them. So these multiple rootlets are the ones that join to form a nerve root. We have the dorsal rootlets here, which join to form the dorsal roots. We also have the ventral rootlets there that join to form the ventral root. And so eventually, a spinal nerve has a dorsal root, which has a sensory function, and a ventral root, which has a motor function. But remember, they come from rootlets. It's a series of rootlets. Having said so, a whole spinal nerve is a mixed nerve with both sensory as well as motor functions. This other image shows you the same concept, but now the ventral side is the one in front here. So this is the ventral root, that's the dorsal root, and there are rootlets there. Point to note is that the dorsal root of the spinal nerve usually has a swelling, which we call the dorsal root ganglion, containing the cell bodies of the sensory nerves. Maybe when we are at this image, it is one of the best time to talk about the components of a spinal, basically the components of a reflex arc. It doesn't have to be spinal. So what are the basic components of a reflex arc? A reflex arc has the following components. One, there must be a sensory receptor that detects the stimuli. Once the sensory receptors detect the stimulus is relayed to the central nervous system through the afferent nerve. And so the afferent nerve is the sensory nerve. The sensory nerve terminates in the integration center. The integration center here can either be a brainstem or a spinal cord. So it doesn't have to be a spinal cord. It could be also brainstem. But in this case, we have the spinal cord. From the integration center, we have an efferent nerve, which is the motor neuron. And from there, we have the effector. The effectors are usually muscles in this case. So those are the basic components of a reflex arc, just in case. All right, let's talk about the spinal nerves in great detail. So there are 31 pairs in total. We know that 8 pairs come from cervical region. We name them C1 to C8. 12 pairs from the thoracic segment. That's how we name them. 5 pairs from lumbar segment, 5 pairs from sacral segment, and only 1 pair from the coccygeal segment. Because the spinal cord has hung upwards or on the upper part at level of L1, L2 in adults, the lower nerves will come out first in a vertical orientation before they exit through their respective segments. It is for that reason then we have the existence of the cauda equina, which we know. Great. So if we've understood that, then we know why there are enlargements as well. The enlargements of the cord are regions where the spinal cord are relatively swollen. And this is because there are so many nerves coming out from those regions. Why would so many nerves come from those regions? Because these regions are related to innervation of the limbs. So remember that the limbs are touched on specific segments of the trunk. And so the nerves that come from those regions will tend to be more because there are more tissues to be innervated in those segments. The cervical enlargement therefore gives rise to the brachial plexus for innervation of the upper limb. And the lumbar enlargement gives rise to the lumbosacral plexus for innervation of the lower limb. So I think what I've given you so far is a brief topographic anatomy of the spinal cord that I wanted us to highlight first before we now start the histological structure. Now, in terms of histology, what we had already said earlier in the previous lecture that you may have listened to is that the internal of the spinal cord itself first is surrounded by meninges, just like the brain. So we have dura mater, pia mater, and arachnoid mater. There's some differences between the spinal dura and the cranial dura. You may want to check on that. The diameter of the spinal cord varies a lot depending on the segment. Now, the concept is that the upper segments will tend to be wider compared to the lower segments. That's one. But also the regions of the enlargements will tend to be larger than the other regions. So consider that understanding. The upper segments are wider than lower segments, but also regions of enlargements are significantly larger compared to the regions without enlargements. The shape of the spinal cord also varies, largely rounded, although the cervical cord will tend to have an orientation that appears ovoid, shorter anteroposterior and wider transversely, but largely rounded orientation. There's some key sulci that help you to know which side of the spinal cord you're looking at. And two important ones to mention. The ventral median fissure or sulcus is a very deep sulcus of the spinal cord. And as the name suggests, it is ventral in front and median, it's in the middle. And so it separates right and left. The presence of that one tells you which part of the spinal cord you're looking at. So the moment you see that, we know that this is the front side. Therefore, this is the posterior side by default. The other fissure to look at is the dorsolateral sulcus, which is this one. The dorsolateral sulci are paired, so they're not midline, but paired right and left dorsolateral sulcus. Again, they are dorsolateral. Now, they're a little fainter compared to the ventral median fissure, as you can see here. And they're paired as well. So you can use that concept to orient yourself. Even if this part was facing whichever direction, it will still be the anterior part of the cord. Dorsally, there's a small sulcus, which we call the dorsal sulcus, very faint compared to the dorsolateral. The dorsal sulcus is very faint. And extending from the dorsal sulcus is what usually called the dorsal septum. There's a septum-like structure in this region that separates, again, the right and the left side of the spinal cord. Now, we can divide the parts of the spinal cord into three major regions. We have this one, which we call the central canal of the spinal cord. Then we have this region, which we call the spinal cord gray matter. As you can see, it's butterfly-like or letter H-shaped around the central canal of the spinal cord. And then the third region is the spinal cord white matter, which is the outer region of the spinal cord. So generally, the internal structure has those three major parts. The relative sizes and configurations of the spinal cord gray matter and white matter vary a lot according to the level of the spinal cord. And that's one of the concepts you're going to use when you're going to basically classify or try to pinpoint the segment of the spinal cord that one is coming from. Right, so we'll be talking much about that. Let's talk about the spinal cord ependymal lining. So the spinal cord ependymal lining surrounds the central canal of the spinal cord. This is the central canal of the spinal cord. The image displayed to you here is the image of the spinal cord. The central canal itself is very tiny. Although it's tiny, it extends the whole of the entire length of the spinal cord actually, and it has cerebrospinal fluid inside it. It could be very tiny to the point that you may not sometimes see it, and that's why I'm mentioning it. So when you look at slides of the spinal cord, sometimes you may not see that central canal easily because it's very tiny in some regions. Well enough, we can see it in this one. So proximally, this central canal goes through the lower part of the medulla oblongata, which means that the lower part of the medulla oblongata also has a central canal. But the upper part of the medulla oblongata does not have the central canal. That means that there's a part of the medulla that encloses the central canal, and there's a part of the medulla that does not enclose the central canal. Now, the part of the medulla that does not enclose the central canal, the upper medulla, is the one related to the fourth ventricle. And so this central canal empties into the fourth ventricle. That makes us understand, therefore, that the medulla has a lower part, which we call the closed medulla, and an upper part that we call the open medulla. We'll talk much about that concept when you look at brainstem histology. In particular, the central canal of the spinal cord is lined by the ependyma. The ependyma is basically a simple columnar ciliated epithelium. Let me show you a high magnification of that. So this shows you simple columnar ciliated epithelium. Okay, at this point, you may only appreciate that it's a relatively simple columnar epithelium, but maybe that magnification displays to you the cilia itself, although not very prominent, but it's there. So simple columnar ciliated epithelium is the epithelium of the ependyma of the spinal cord. In some situations, this epithelium may be cuboidal, but no big deal. Most of the time, it's ciliated columnar. What's the role of this epithelium? It will secrete CSF into this space. But other than secretion of cerebrospinal fluid, the ciliary activity of the ependyma promotes the flow of the cerebrospinal fluid. That's it regarding the ependyma. Now let's talk about the spinal cord gray matter. The spinal cord gray matter is a complex mixture of cell bodies of neurons, the processes of these neurons, their synaptic connections, as well as some blood vessels and supporting cells of the nervous tissue. So it's a complex of all of them. However, the definition of it being gray matter means that there must be cell bodies. And so the presence of the neuronal cell bodies is what makes it constitute as gray matter. And remember, gray matter of nervous tissue may have different terms depending on where that gray matter is found. If the gray matter is in the peripheral zones of the brain, then it constitutes what we call the cortex. And we can have cerebral cortex, we can have cerebellar cortex. But if that gray matter is in the deep substance of the central nervous system, then it constitutes what we call a nucleus. And so by default, the spinal cord gray matter is a nucleus or a collection of different nuclei, as we're going to see. It is organized into two major horns, but fine, there's usually a third horn. So let's say it has three horns. We have the ventral horn, this region here, which is also called the anterior horn. This is the region that gives rise to somatic motor neurons. These are the neurons that innervate skeletal musculature. So the cell bodies of the lower motor neurons, as we call it, are housed within this region. It has a somatic motor function. Then we have the dorsal horn, which is also known as the posterior horn. The dorsal horn or the posterior horn receive sensory nerve terminals. They enter there. Although, as we're going to see, that could not be the only function of the dorsal horn. The intermediate horn is not necessarily present in the whole of the spinal cord. It is only present in some regions. So which regions of the spinal cord have the intermediate horn? From segment T1 to L2 or LT3 sometimes of the spinal cord, and segments S2, S3, and S4. Only those have the lateral horn. And the lateral horn is for autonomic efferent. It houses the cell bodies of preganglionic autonomic neurons. So that's the general structure of the spinal cord gray matter. Now I want to practically take you through some concepts of the spinal cord gray matter. So this is how it will look like. If you look at a slide of the spinal cord, that's how it will look like. There's some points to note. That this spinal cord gray matter, we've said that's butterfly-like, or letter H. That makes us understand that there's some 4 linked cellular masses. So the 4 linked cellular masses are largely the ventral horns and the dorsal horns, because I've told you that the lateral horn is not necessarily present in all of them, and when present, again, is very tiny. So the major concept here is that there are 4 linked cellular masses, the paired dorsal horns and the paired ventral horns. Now take note of the gray matter around the central canal. The right and the left dorsal horns are continuous posterior to the central canal. We call that continuation, the dorsal gray commissure. Similarly, the right and the left ventral horns are continuous ventral to the central canal. We call that continuation the ventral gray commissure. Don't confuse the gray commissures with the white commissures of the white matter. So here, this is the dorsal gray commissure, and this is the ventral gray commissure. Maybe you don't know what commissure is. A commissure is basically a connection between the right and the left side in this context. Now, when you look at the gray matter, we see cell bodies of neurons. the type of neurons which are present within the spinal cord gray matter are multipolar neurons. So don't open your mouth and mention about pseudonipolar neurons and bipolar neurons in the spinal cord gray matter. They are multipolar neurons. Whether they are motor or they are sensory, doesn't matter. They are multipolar neurons. So these are the cell bodies as you can see them. And this image shows us something else other than the cell bodies. I want to use this image to show you something. That the gray matter of the spinal cord is really heterogeneous. We can see variations in the sizes of the neurons. Some neurons are tiny, some are very large, as we can see here. And we can see some zonations. I'm coming to that shortly. Now, let's follow the dorsal horns posterolaterally toward the surface. We can see that the dorsal horn and the tip of the dorsal horn there towards the surface has something there. So this region here is unique. We call it the dorsolateral tract of Lissauer. The dorsolateral tract of Lissauer is not part of the gray matter. It is not part of the dorsal horn, but the route through which white matter, axons of neurons, pass as they enter the dorsal horn of the gray matter. And so they constitute the dorsolateral tract of Lissauer, that region and that region. I want also to mention to you something on the anterior horn, the anterior horn cells. Now look at the anterior horn of the spinal cord gray matter and note that the cells, the multipolar neurons in this region are predominantly larger compared to the other regions. We call these cells the anterior horn cells, or the ventral horn cells of the spinal cord. This is how they look like at a high magnification. They are multipolar neurons. They function as lower motor neurons of the spinal cord. Now, if you listen to the lecture on introduction to anatomy of the central nervous system, then you know what lower motor neuron is and upper motor neuron is. All right, so maybe I use this opportunity also to tell you something about neurons. There's some characteristic histological features of neurons that you're able to pick. And these neurons display them. Not all neurons, but some neurons have these features. You may have a very large nucleus. You're looking at that, you know, the whole of this is the cell body. And so this large light region here is actually the nucleus. It might be tempting to think it is part of the cytoplasm, but no. This is the nucleus. And if that's the nucleus, that central dot is actually the nucleolus. It might be tempting to think that that central dot is a nucleus, but no, that's the nucleolus, and this is a nucleus. So what do we see? These neurons have a very large nucleus that is lightly staining. What term do we use for lightly staining neurons or lightly staining nuclei? We say they are euchromatic, which means they contain euchromatin within them. So they are large euchromatic nuclei with prominent nucleolus. That kind of a cell is described as open-faced nucleus. An open-faced nucleus represents a nucleus that is consistent with active protein synthesis and secretion. And so we see open-faced nucleus or vesicular nuclei in cells which are actively involved in protein synthesis, and neurons are one of them. Now let's talk about what we call the Rex laminae of the spinal cord gray matter. The spinal cord gray matter can be described into 10 different histological layers or 10 different laminae. These laminae are termed the Rex laminae. The distinction of the laminae are based on a number of concepts, largely based on the size of the neurons within that particular population, the shape of those neurons, the cytological features of those neurons, and even their density. How packed are they? Now, it's not your role to redefine them, but basically to understand what is where. With that in mind, I want you to know the following: that they are numbered from number 1 to 10 in a dorsoventral sequence, which means we start from this side, the dorsal side, as we go that way. It's a dorsoventral sequence, largely. With that in mind, we have lamina 1 to 6. Lamina 1 to 6 is the one representing the dorsal horn. Dorsal horn has lamina 1 to 6 in that order. Now, in particular, lamina 1 to 4 are the ones which represent the peripheral parts or the tip of the dorsal horn, the elongated tip of the dorsal horn. This lamina 1 to 4 is the main site of termination of first-order sensory neurons. The first-order sensory neurons are the neurons which enter the spinal cord from their peripheral receptors. So lamina 1 to 4 are the main site of termination of the first-order sensory neurons. Histologically, in lamina 1 to 4, there are 4 different layers of cell populations, and that's why they are named 1 to 4. And you may come across some names. Lamina 1, which is the most peripheral, is the one we call the lamina marginalis, or the marginal zone. Lamina 2 is slightly bigger. We call it the substantia gelatinosa, or the gelatinous substance. Largely called so because of the neurons in that particular region do not necessarily have myelin sheaths. And so when we use myelin stains to stain the spinal cord, that region appears clear. They don't take up stain. And so it appears gelatinous. Lamina 3 and 4 constitute what we call the nucleus proprius of the posterior horn. So that's lamina 3 and 4, nucleus proprius of the posterior horn. Generally speaking, lamina 1 to 4 is a point of termination of the primary sensory nerves. Then we have lamina 5 and lamina 6. They represent the base of the dorsal horn. That's lamina 5 and 6, the base of the dorsal horn. So this lamina 5 and 6, which represent the base of the dorsal horn, receives proprioceptive impulses. Proprioceptive impulses are impulses that determine position. So their sense of position, proprioception, the sense of position. So impulses that determine position, those are proprioceptive impulses. It receives that one. But in addition to receiving that one, it also receives some descending motor projections, maybe from the cerebral cortex. We call that corticospinal. Because of that connection, it's receiving impulses from the cerebral cortex, which are motor in function, It's also receiving impulses from peripheral receptors for proprioception. Lamina 5 and 6 are known to regulate movements. Okay, that is about the dorsal horn. Then you have lamina 7. Lamina 7 is slightly bigger, but corresponding with the level of the intermediate horn of the spinal cord. It's a very extensive zone with heterogeneous cell populations and therefore also different functions. Within this region, we have the well-known thoracic nucleus of Clark, largely present in the thoracic spinal cord, T1 to, may I extend up to L3, but mainly in the thoracic column. So it's called the thoracic nucleus of Clark's. Among other nuclei present here, you can see the intermediolateral and the intermediomedial nuclei or regions there, also part of lamina 7. The main function of lamina 7 is to regulate posture and movement, and that's largely the role of the thoracic nucleus of Clark, and also to regulate autonomic functions, and that is largely the role of the intermediolateral and intermediomedial nuclei. In particular, the intermediomedial nuclei is responsible for sympathetic functions, and the intermedial nuclei is responsible for the parasympathetic nuclei functions. So what I want you to understand is this, that lamina 7 houses cell bodies of preganglionic autonomic neurons. And those ones are particularly present on the lateral horn. So you'll only have those ones in segments of the spinal cord, the lateral horn, T1 to L3 or L2 and S2 to S4. S2 to S4 is parasympathetic, T1 to L2, L3 is sympathetic. Other than that, the lamina 7 also has other functions in terms of regulation of posture and movement. And so there are some pathways that relay within this region as they ascend, but they're responsible for posture. Then we go to lamina 8 and 9, which are largely present in the anterior horn. These are the ones that are exclusively or particularly involved in somatic motor functions. In particular, lamina 9 is the one that houses the cell bodies of the lower motor neurons. The ventral horn cells of the spinal cord are housed by lamina 9. Lamina 10 represents the gray matter around the central canal of the spinal cord. And so we can call it the pericentral gray matter. It therefore includes the ventral gray commissure and the dorsal gray commissure of the spinal cord. All right, so that is the organization of the spinal cord gray matter. Now let's talk about the organization of the spinal cord white matter. I'll not talk much on the organization of the spinal cord white matter, largely because it involves the understanding of what we call tractology, pathways. And I believe there's a better lecture to highlight on the spinal cord tracts. Multiple lectures for that matter, ascending, tract, descending. So I'll just say something brief. The spinal cord white matter contain nerve fibers, which are largely axonal processes. These axonal processes can either be carrying impulses upwards, so that means they're ascending, hence termed sensory, but they could also be carrying impulses from the brain downwards, then termed descending or motor. structurally, some of these axons are very long. They go beyond the spinal cord. So we call them the long tracts. Now, what are tracts? We already mentioned a group of axonal processes of the same function called a tract. So they could be long tracts, but you can also be having the short one. The short ones come from one segment of the spinal cord to another segment of the spinal cord, and those short ones are termed propriospinal tracts. The propriospinal tracts can still either be ascending or descending. We divide the spinal cord white matter into three major columns, or what we call funiculi. We have the anterior funiculus, the lateral funiculus, and the dorsal funiculus. This image still captures that, the anterior funiculus, lateral funiculus, and the dorsal funiculus. In this slide again, so this is the white matter. So this is the posterior white column. This is the lateral white column. And this is anterior white column. As you can see, they merge into one another, the anterior and the lateral one. The lateral and the dorsal one are separated by the dorsolateral tract of Lissauer there. The right and the left dorsal funicula are separated by the dorsal septum. The anterior white column are joined by a commissure here, which we call the anterior white commissure, joining the right and left white commissure, or right and left white matter. Important to note is that the spinal cord white matter contain tracts, and so I've just put this image here, not for discussion, but to help you understand or to trigger your understanding that there are several tracts within the spinal cord white matter. Some of them are going up, called ascending, carrying sensory information. Some are going down, called descending, carrying motor information. I don't feel like this is the correct lecture to talk about the various tracts of the spinal cord, but you may have listened to that in the long lecture that I gave. That's good enough. There are still specific lectures of the spinal cord that focus on tracts. There are lectures that focus on ascending tracts, and there are lectures that focus on descending tracts. But in general, look at the posterior white matter. The posterior white column contains two major tracts. We call it fasciculus gracilis and fasciculus cuneatus. Fasciculus gracilis carry information from the lower part of the body. Fasciculus cuneatus carry information from the upper part of the body, but they're all in the dorsal column. I want you to take note of that because we are going to use it shortly. In the lateral side, in the lateral white matter, we have these tracts which are in the periphery there. We call them the spinocerebellar tracts. But deep to that, we have some descending tracts, in particular, the one that come from the cerebral cortex to the spinal cord, corticospinal, which is a descending tract. Then coming down, you can see many other tracts there. So there'll be another lecture that focuses on tractology. Great. The last thing we are going to do is now practically go through different histological segments of the spinal cord and try to know which segment of the spinal cord that is. So this is what I'll do. I'll project for you a chord segment, and I'll want you to think about that chord segment. Just give it a segment first, and then I'll make a conclusion. So I have a series of 10 slides that you're supposed to comment on. Remember, you're identifying a chord segment. Before that, maybe I give you some tip. The tip is this. On the spinal cord, we know that the spinal cord, when you see some things, one, when you see the characteristic shape of the spinal cord gray matter, very important to note. We also know that the spinal cord, because it has a central canal which is lined by ependyma. But remember what I mentioned, that central canal may not be necessarily visible in all slides. So it may not be a very strong point to use. But when present, when you are able to see the canal, then remember that canal is lined by ependyma and it's surrounded by the gray matter. Very important to note that if present true, it should be within the gray matter lined by ependyma. Third point to note is that the region outside the gray matter will appear largely fibrous because it contains the tracts. Last but not least, very important that you orient yourself on which side is in front, which side is behind. And to help you do this is your ability to identify the ventral median fissure or ventral median sulcus. Once you've identified it, then you know which side is in front, which side is therefore not in front. With that in mind, what do we use to know a chord segment? You'll use the following. You'll use the relative size of the white matter. You'll also use the absolute size of the white matter. Now, white matter will be more in the upper chord segments compared to lower chord segments for a simple reason. The upper chord segments has all of them, or has already received or yet to give out all of them. But the lower chord segments will be lacking them because a number have already terminated in various segments. And so when you look at a spinal cord section and you see a lot of white matter, your conclusion is that it's upper regions of the cord rather than lower regions of the cord. For that reason, a cervical cord will have a lot of white matter compared to a lumbar cord or a sacral cord. But understand the concept. So when it's a lot, doesn't necessarily mean cervical. You know, even T1 will have a lot compared to T12, they're all thoracic. C1 will have a lot compared to C7, they're all cervical. It therefore requires that you train your eye to be seeing the spinal cord and determine what exactly is a lot. And this is important also to be comparing the gray matter in that region. The next thing that you need to know, and very important, is the relative size of the gray matter zones, in particular, the relative size of the ventral horn compared to the dorsal horn of that particular segment. That has a lot of implications. But as a general rule, the ventral horn is usually larger than the dorsal horn, as a general rule. And so again, you need to be training your eye to know what exactly do you mean by large. However, there are some regions where the transverse diameter of the ventral horn is quite pronounced compared to the transverse diameter of the dorsal horn. Those regions are regions which give rise to the plexuses that supply the limbs. I've just mentioned to you that the ventral horn cells are quite big. And so if many of them are arising from a constricted zone, then we'll expect the gray matter of that region to be expanded. And that's what happens here. That is why the regions that give rise to plexuses, because they're supplying a lot of skeletal musculature of the limbs, their ventral horns are quite prominent. That does not mean that their dorsal horns are not prominent compared to the other segment. They're still prominent, except what you're saying, that the prominence is more on the ventral horn than the dorsal horn. And so when you compare those ratios, you'll see that the ventral horn is almost twice or 3 times larger than the dorsal horn. That region is giving rise to a plexus. Now think about that and ask yourself, so which region give rise to the brachial plexus? It will come from around C5 up to T1 segment of the spinal cord. So you'll expect a T1 also to have a prominent ventral horn, trust me, because it gives rise to a plexus. Generally speaking, therefore, we'll expect this prominent ventral horn to be in the lower cervical cord, mainly. But we also expect that to be the truth for the lumbosacral plexus, or the region given rise to lumbosacral plexus, which refers to the region from around L1 up to S3. So L1 to S3 segments of the spinal cord also have a prominent ventral horn. Then we need to check the presence of the intermediate horn, the presence of the lateral horn. I will admit it's very difficult to see the lateral horn in most slides. But when you see it, you will know it is the one. It's usually very tiny and sharp-like, not so much prominent, and slightly posterior or in the middle there at the level of the central canal. When the intermediate horn is present, it tells you something very unique. It tells you that this segment of the cord gives rise to preganglionic autonomic neurons, because that's the role of the intermediate horn. So only segments T1 up to L2 or L3 will have this horn for the sympathetic outflow. And segments S2, S3, and S4 will also have this horn for the parasympathetic outflow. All the other segments of the cord will not have the intermediate horn. The other thing, so the first 3 things that I've mentioned are the most important things to check, and that will help you identify chord segments by all means. But the other 2 that I'll add to you, you may use them, not all the time. So you may want to look at the dorsal column. And in the dorsal column, we are looking at 2 concepts. The volume of the dorsal column, how big is it? And we're also looking at, is it partitioned? In terms of volume, if the dorsal column has a lot of fibers, that can only mean that you're on the upper part of the cord. So almost a similar concept like point number 1. If the dorsal column has thin fiber system, it simply means that you're in the lower part of the cord. So that is with regard to size. Again, that requires that you train your eye to know what is a lot and what's little. But there's something also important to note. The partitioning of the dorsal column. Is the dorsal column split? Now, I told you that the dorsal column has fasciculus gracilis and fasciculus cuneatus as tracts within it. If the dorsal column is partitioned into those 2, it tells you that we have both fasciculus gracilis and fasciculus cuneatus present. And that can only mean one thing. We are on the upper parts of the spinal cord. Because I told you that gracilis carry information from the lower part of the body and cuneatus from the upper part of the body. So you cannot expect cuneatus to be present in the lower part of the spinal cord. but gracilis will be present in the whole of the spinal cord because it carries from the lower part going up. Generally speaking, pinata starts to be existent from T6 upwards, so T6, T5 upwards. That does not mean, again, that you easily see cuneators at T5, T4, not quite, because it's too small to be distinguished. So what am I saying? You can only use this concept if the partitioning is actually present, then you are very sure of what you are saying. But if the partitioning is not present, you cannot use this point as a determining factor. The absence of the partitioning does not necessarily mean that it's the lower part of the cord, because sometimes the partitioning is not prominent. But the presence of the partitioning definitely tells you that we have cuneators and gracilis. I hope that's clear. The last thing to point at the number of collateral spinal nerve roots that we see running on the outside of the spinal cord. Now, you need to remember that there's something called the cauda equina. This is because the spinal cord has shortened, and so the lower nerve roots take a vertical orientation before they exit. If the lower nerve roots take vertical orientation before they exit, if you take sections of the spinal cord, the lower part, it is possible to see several nerve roots on the outside because we are taking a vertical orientation. It is possible to see. And so when you see several nerve roots in the outside, collaterals, it simply means that we are around or very near the cauda equina. But if you see few, if we see few, it may not tell you much though, because of two reasons. When we section the spinal cord, we do not necessarily retain the collateral. Sometimes a spinal cord section may have been obtained and the collaterals were removed. So again, this is one of the points I want you to use it only when it's present. When you see several collaterals, then we are definitely sure it's the lower segments of the cord and not upper segments. But when you don't see the collaterals, you can't say it is lower or upper because maybe they were removed during the processing of the tissues. Great, so these are the concepts we are going to use to identify the 10 slides that I've told you. So I hope you are ready. I'll be quick on those slides because now we've mentioned much of the things. Now let's begin with this slide. So that's the first slide. I will put it to you to give a comment on the chat. Just state what you think. 30 seconds you have. Put in the chat. All right, good. I have seen most people have gotten that right. Actually, almost everyone who has responded. This is not the spinal cord. It is part of the brain stem. However, I have seen many are saying medulla. It is not medulla as well. It is midbrain. We'll talk about it in another session. Okay, slide 2. Again, just put it on the chart there. Good enough? Again, not the spinal cord. Now, why are we saying it's not the spinal cord? We don't see the butterfly. We don't see the ventromedian fissure. It is not the spinal cord. And that also applies to this one. It is not the spinal cord. This one and the previous one are the same. This is pons. We'll talk about in another session. That also applies to this one. We don't see the butterfly. We see something else, but not the butterfly. This is the upper part of the medulla oblongata, the one that is open. So we call it open medulla. This one is also not spinal cord, in as much as you might be tempted to think so. There is no butterfly. This is closed medulla. Again, this could be quite tempting for spinal cord, but it's not. There's no butterfly. This is still closed medulla. So the two medulla I've shown you for closed, this is called medulla at motor decussation, and the previous one, medulla at sensory decussation. We'll talk about them more in other sessions. Now I'm showing this slide of cervical cord. What do we note? A lot of white matter present. Also, we note that the posterior column is partitioned into fasciculus gracilis and fasciculus cuneatus. We also note that the anterior horn, the transverse diameter of the anterior horn, is quite prominent compared to that one. That tells you that it's more of the lower cervical cord rather than the upper cervical cord. It's giving rise to brachial plexus. Something important to note about cervical cord is that the anteroposterior dimension is shorter compared to the transverse dimension. That is cervical cord. I'm showing now the slide of thoracic spinal cord. Why do we say so? Important things to highlight is the presence of the lateral horn. It is present. But why not, therefore, can we say this is sacral? Because the white matter is still quite a lot as well. There's more of upper parts of the cord rather than the lower parts of the cord. Could it be a T1? Maybe not. Because the anterior horn here, the transverse dimension here, and that one, yes, the anterior horn is larger, but not quite large compared to what we saw. So it's more of typical thoracic spinal cord. You can see a few collaterals, that one and that one. So these ones are few collaterals, they're not a lot. The most important thing for you to note about the thoracic cord is that the presence of the lateral horn and the anterior horn and dorsal horn, which are almost the same size. Of course, this is a ventral commissure, so you know this is the anterior side. I'm now showing you a lumbar cord, and we see a lot of collaterals. So we know that we are near the cauda equina. They are present at least in this one. Now look at the white matter. It has reduced compared to the previous slide. But look at the gray matter. We can see gray matter is a bit prominent really because the white matter has reduced. So these are lower parts of the cord. This is particularly lumbar cord. And lastly, this is sacral cord. The gray matter is almost filling the whole of the spinal cord. White matter is thin, very very tiny. We can see a few collaterals there. Remember, I told you something about the presence and the absence of them. So when there are few or none, doesn't tell you much, but when a lot tells you something. The most important thing to note in this slide is that the gray matter is so prominent and white matter hardly there. And especially look at the dorsal column. Very thin. This is sacral cord. Almost lacking white matter. We may not be able to see nicely whether there's a lateral horn or not, so we don't have to speak much about it. Great, so those are the concepts used to identify spinal cord, and I hope now that you are well oriented in spinal cord histology. It will be important to take time to understand much of the spinal cord white matter by looking at the lecture on ascending tracts of the spinal cord and brainstem.